Great. We're recording the session today, so let's wait couple of minutes till people get to join us. We are very grateful because we know today is Monday, so it's a big exception for us. So we are very grateful for everybody who can join us today. Yes, please remember that recording the session, it will be done by us, but should not be done by you because many data is unpublished. Rosa, you wanted to say something?
Yeah, because in the chat there was this message of AI notes.
AI notes is not accepted.
So I don't know, this LPJ lab, if you can stop this recording on the side. Thank you.
Yes. So, and we also realized there's been the daylight saving time. So some people join earlier, some people may join too late. So hopefully this will be okay. So it is a pleasure for me to introduce our speaker today. It's Sarah Marzi. I'm just gonna share my screen to introduce the team. Okay. You know, this has been organized by the University of Lausanne, the team of Rosa Paulicelli with Anne-Claire Compagnon, who will handle the questions and answers, Gloria Colombo, who will do the introduction, Giorgio Corsi and Felipe Spinoza, from the Achucaro team, myself, Fede Soria, and Marta Pereira. We anticipate that there will be some changes in the organization in the coming months, so stay tuned because it's going to be very exciting. So without further ado, I'm very grateful that we had Sarah Marzi joining us today from UK Dementia Research Institute at King's College. So Gloria, go ahead, please, with the introduction.
Of course. So I'm very happy to introduce today's speaker, Dr. Sarah Marzi, Senior Lecturer at UK Dementia Research Institute, and a group leader at King's College London. So Dr. Marzi's academic journey began with degrees in mathematics and psychology from the University of Freiburg, Germany, followed by her PhD in epigenetics from King's College London in 2017. And she further honed her expertise with an independent research fellowship in neuroepigenomics at Imperial College London, before returning to King's in 2023 to establish her current group. And she's an expert in epigenetic regulation as it relates to human disease. Her lab employs advanced genomic techniques on human brain tissue and model systems, both in vitro and in vivo, combining experimental data with innovative computational analysis to uncover the gene regulatory mechanism that drive the earliest stages of neurodegenerative diseases. So please join me in welcoming Dr. Marzi, and the stage is all yours.
Yeah, just a reminder, if you turn off your cameras, the streaming will be much better. And then we will open up again the floor for the questions and answers later on. Okay, thank you very much.
Right, perfect. Thank you so much for the invitation and the kind introduction. I'm going to assume that everyone can see the screen fine, but if not, please come in.
It's all okay.
Okay, great. So yes, as has been introduced, we're an epigenetics lab that studies neurodegenerative diseases, and we're particularly interested in the earliest events that causally drive disease. And we do this by studying how genetic risk factors and environmental risk factors drive epigenetic regulation in disease-relevant cell types, and thereby make cells vulnerable and predispose them to disease. We're not, to begin with, a specifically microglia-focused lab, but in a lot of our research, both in Alzheimer's and Parkinson's disease, the data has repeatedly led us to patterns and signatures epigenetically that are critical in microglia. And so I want to tell you a few of these stories today and how some of this gene regulation is sort of converging on pathogenic and potentially also protective microglia regulatory patterns and networks. So as I said, our main motivation for studying epigenetics is that we want to understand the really causal onset risk factors. So the genetics, the environmental factors, but also there's those things that we can't necessarily change that happen to everyone, but that we know contribute to vulnerability and disease risk, such as aging. So we all age, but we do know age is the single biggest risk factor for Alzheimer's disease and other late onset neurodegenerative diseases. And we want to interpret these kinds of risk factors and what they do to our cells by studying epigenetic regulation. Why is this sensible and important? Well, it's been shown by many, many studies that the genetic risk associated with most complex neurodegenerative diseases, and the example here is the genome-wide association study of Alzheimer's disease from 2022. The majority of those risk variants fall into non-coding regions of the genome, and so they don't really change protein sequences and functions themselves, but rather they tend to fall into regulatory elements that then alter the expression of genes downstream. And so we can study the epigenome to interpret in which cells, in which contexts this happens, what the downstream targets are, and what the affected biological mechanisms are. On the environmental side, the example here is from a review we wrote on Parkinson's disease, and these are some of the, or actually all of the reported risk factors in the human epidemiological literature. And so the question that we really aim to ask here is, how can these risk factors, to which oftentimes we're exposed at very, very low grades chronically over many years and sometimes even decades, how do they then much, much later cause these very specific disease syndromes? And so we think that epigenetics in this case provides a cellular memory substrate that can retain information from these chronic low-grade exposures, and that sort of gradually alters the regulatory potential and patterns to drive them into states of vulnerability or predisposition to disease. And in fact, we do know from some human work and a lot of really good model system work that a lot of these reported risk factors, in fact, do have effects on the epigenome and can alter epigenetic patterns. And likewise with aging, in fact, aging is so strongly associated with epigenetic mechanisms that you can just look at the epigenome, for example, the DNA methylation of a person in any cell type or tissue in their body, and very accurately predict how old that person is. And so there's this direct link and really a need to understand what these aging-related epigenetic changes are and how they relate to disease vulnerability. Now, when I talk about epigenetics, I use quite a functional biological definition, and that would be any biochemical mechanisms that regulate gene expression, and they should ideally in dividing cells be heritable. So when the DNA is copied over, the epigenetic mechanisms are also copied over, or in post-mitotic cells, they should at least have some degree of little bit of stability. So we're not talking about things that happen at less than a second or few second range. We're talking about slightly slower mechanisms that alter gene regulatory potential. And practically, this is three classes of mechanisms. So we have DNA modifications, and in humans and other mammals, this is most prevalently in the form of DNA methylation. Then we have modifications to the histone proteins around which the DNA is wrapped in the cell nucleus. And these can either attract or repel DNA, thereby making it accessible or closing it off to interaction with transcriptional machinery. And there are many, many different histone modifications. These can also be things like methylation or phosphorylation, acetylation, ubiquitination, you name it. We keep discovering more and more different ones, but they tend to occur not completely at random. They occur in very specific patterns that define regions of the genome that really have specific functions, like transcription start sites and promoters will have very, very specific combinations of histone modifications. Other regulatory elements like enhancers will, and then heterochromatin looks different again, gene bodies look different. And then the third class that I don't really work on, but just for completion is worth mentioning, are non-coding RNAs. So these are RNAs that are transcribed but not translated into protein. Rather, the RNAs themselves have functional roles, such as microRNAs, for example, which are able to post-transcriptionally regulate other RNAs by cutting down transcripts, for example. And a lot of my research really focuses on enhancer-driven gene regulation. So enhancers are regulatory elements that usually sit upstream of the genes that they regulate, and they can be in close vicinity to that target gene. They can also, along the genome, be very, very far away, maybe a megabase away or so. But the way they regulate their target gene is that in 3D space within the nucleus, they fold onto the promoter of the target gene, as you can see here, enhancer interacting with a promoter. And these interactions are thought to be primarily driven by these two proteins, casein and CTCF. And then there are a number of other molecules and factors involved, like transcription factors, chromatin modifiers, mediator complexes. But that's the basic principle of how enhancer-driven gene regulation works. And one of the most critical enhancer marks, histone modification that is found at active enhancers, is H3K27 acetyl. So this means histone 3, lysine 27 acetylation. And this is really the modification that I've been studying for the past many, many years, over 10 years now. And it's found exclusively at active promoters and active enhancers. It's quite cell type specific, and that's because enhancers are very cell type specific. It's quite dynamic in response to environmental risk factors, and it also, as I'll show you in a minute, relates really very much to the genetic risk of these complex neurodegenerative diseases. So a number of years ago now, we profiled this exact modification, H3K27 acetyl, in post-mortem samples in the human entorhinal cortex. And we showed that the genetic risk for Alzheimer's disease, but also a number of other neurological diseases like ALS and psychiatric diseases like schizophrenia, is really enriched in the regions that are marked by H3K27 acetyl. And you do this with a method called linkage disequilibrium score regression, but essentially you ask, based on the number of genetic variants that fall into your regions of interest, so into those regions that are marked by H3K27 acetyl, how strong are the effects, so the association with the disease at those variants, compared to the rest of the genome. And if it's enriched in those variants, you basically get a significant result there. And it turns out that it is really enriched, and it's specifically enriched in the brain and not in other tissues. But since then, and with other methods, we actually know it's not just the brain generally, but it's actually in Alzheimer's very consistently the microglia that come up. And I'm just showing two examples here of studies that were led by collaborators of mine, Nathan Skeen and Alexi Knott, who actually now are both at the UK Dementia Research Institute at Imperial College London. But so Nathan basically worked on mouse single-cell RNA-seq data and showed that the genetic risk for AD is specifically enriched near genes that are only expressed in microglia, or most specifically expressed in microglia, whereas Alexi worked on human resected tissue and profiled different histone modifications, including H3K27 acetyl. And with his epigenetic data in human, he was able to show that the genetic risk for AD is enriched in microglia, and not just anywhere in microglia, but it's specifically the enhancers. So really, if we want to interpret that genetic risk and understand what it's actually doing and how it's predisposing our brains to disease, we need to be studying microglia, and we need to be studying microglial enhancers. One way in which you can then more globally interpret the risk of AD is by sort of aggregating the genetic association signal into the likely causal genes that are driving these associations. And there's a method called MAGMA that does just this, but one of the challenges is actually annotating the variants to the genes that they're impacting on. And in the same way, it's challenging to annotate an enhancer to the gene that it's regulating. Sometimes it's the closest by gene, but not all the time. As I told you, they can fold in 3D space over quite long distances onto their target promoter. And these 3D interactions can best be captured by methods that profile chromatin interactions, such as Hi-C. And so by integrating Hi-C with this MAGMA approach, you can basically take variants, annotate them to their chromatin interaction defined gene, and then aggregate all the evidence from the genome-wide association study at the gene level. So how much evidence for the causal involvement for a gene based on the GWAS signal is there? And this method was adapted by Hye-Jung-Won's lab a few years ago. And so actually the first project I'm telling you about is a work led by Alexei Nott's lab that I was a little bit involved in, and it was led overall by a PhD student that we are co-supervising, Aidan Askarova. And so Alexei had these beautiful cell type specific epigenetic profiles and also cell type specific chromatin interaction data sets. And so he used that to apply HMAGMA and to really interpret what the causal genes and pathways are that are implicated by genome-wide association studies in AD and other neurodegenerative diseases. And so overall, what he found is that indeed, that again, this study confirms that for AD, the genetic risk is really specifically enriched in the microglia. It's also the microglia for multiple sclerosis, no surprise there. And also reassuringly for diseases, generally for psychiatric and neurodevelopmental diseases, we would expect these to be enriched in neurons and neuronal regulatory loci, and that's in fact also the case in this data set. But more interestingly, actually, is when you think about, so it's microglia in multiple sclerosis, it's microglia in AD. Is it the same kind of genes and processes that are predisposing our brains to these diseases? And so by doing MAGMA, you can then annotate which pathways are implicated by the causally annotated genes. And so when you look at AD, really the most strikingly and most frequently implicated pathway based on the genetics of AD is really lipid homeostasis, lipid processing, lipid metabolism, and that comes up across two independent GWAS studies. There is also some signal for amyloid processing. We would caution about that because that could be slightly circular, because the AD-GWAS, basically genes implicated by AD-GWAS are automatically annotated to amyloid based on being implicated in AD. And so there may be a functional role or it may be a circular argument here. Whereas if you look at other diseases, so for PD, the microglial enrichment was a bit weaker, but for MS, it was really strong. And so when you look at what kind of pathways and what genes are implicated here, really it's very overwhelmingly adaptive immunity. So two diseases here that both have these very clear microglial signatures, but pointing to very, very different pathways underlying the disease risk. Next, I want to move from the global view of disease, and I said the majority of variants associated with Alzheimer's disease are really non-coding variants, and so these aggregatory genome-wide approaches are really helpful there. But we also know that one of the strongest genetic risk factors for late onset Alzheimer's disease is actually the variants in the ApoE gene, and so ApoE occurs in three different forms, ApoE 2, 3, and 4. With the majority of people have ApoE3 genotype, so that's considered sort of the background normal level. And compared to that, if you have ApoE4 homozygously on both of your alleles, your risk for developing disease can be up to 12 times higher than with ApoE3 genotypes. And conversely, ApoE2 tends to be protective. So if you have ApoE2, homozygously in particular, you're very, very unlikely actually to develop Alzheimer's disease. But ApoE it's a lipid processing or a lipid transporter gene, but it's expressed by various different cell types and it tends to have very pleiotropic functions. And so in this work, we really wanted to understand what ApoE does in microglia, and that's because we know from the overall heritability of AD that microglia are very strongly implicated, but we also know that particularly in the brains of people with Alzheimer's disease and in Alzheimer's animal models, that microglia tend to upregulate ApoE very much and start to really express it at high levels. Not so much at baseline, but then when you get neuropathology, and especially the microglia that sort of live around the plaques, express very high levels of ApoE. And so to study what the role of ApoE polymorphisms are, specifically just in microglia and independently of what's happening with the other cells, we teamed up with Bartosz Struper and Renzo Mancuso at VIB in Leuven and Antwerp, and we used the beautiful xenotransplantation model where they can deplete the mouse's own microglia and then inject human iPSC-derived microglia in the mouse brain that then populate the mouse brain and live there, and it can be studied. And so we used an isogenic ApoE series with all the 3 naturally occurring ApoE genotypes, as well as an ApoE knockout to understand the general effect of ApoE. And so this was work experimentally led in my lab by my former postdoc Dihu, and computationally by my former PhD student Kitty Murphy. And what we did here is we transplanted these ApoE series microglia into an Alzheimer's disease mouse model, the APP-NLGF mouse. This is an amyloidogenic model, so it aggregates amyloid, and we kept them for 12 months, and after this time they would have very extensive amyloid pathology, then purified just the human microglia based on human-specific markers, and then did some gene regulatory profiling, so RNA-seq for the gene expression and ATAC-seq for open chromatin signatures. And overall, we found a whole bunch of different changes in terms of transcription. First of all, one thing that stood out because it came up in multiple comparisons was this gene CHCHD2, which is a mitochondrial gene, and variants in this gene have been implicated in Parkinson's disease. And we see that it's downregulated in ApoE4, both when you compare it to ApoE3, and also ApoE2. And when we look at all the 4 genotypes again, it's not just downregulated, it basically isn't expressed in the E4 and also isn't expressed in the knockout microglia. This is also reflected very consistently by the chromatin signature. So this is the CHCHD2 gene down here, transcribed right to left. So upstream here, we're in the promoter region, and you'll see these nice 2 promoter peaks in the E2 and in the E3 genotype, and a complete loss of these promoter peaks in E4 and in the knockout, suggesting that that silencing of transcription of CHCHD2 is driven through epigenetic mechanisms. That was just one single gene example, but often we try to look at a more systematic level, combining multiple genes into co-regulated pathways. And so something that we did with this data was weighted co-expression network analysis, WGCNA, where you cluster basically groups of genes together into a module that behaves similarly across your sample. So if they're always up in sample 1, always down in sample 2. So they have similar patterns of variation across all your samples. And so this is one module that we did identify in our data that was very strongly associated with the different ApoE genotypes. And as you can see, the genes in this module are most highly expressed in E2, medium expressed in E3, lowly expressed in E4. And when we annotated this module, the genes that make up this module, into their biological function, we see that it is really pathways relating to proliferation and migration, both at baseline, but also under immune activated conditions such as chemotaxis, for example. And so what this suggests is that E2 microglia express more genes that promote cell motility and that promote chemotaxis. And so we wondered if they're better at chemotaxis and at getting, in this case, in the amyloid model, to the plaques, are they also better at other microglial functions such as phagocytosing things? And so we took a list of known phagocytosis promoting genes from the literature and checked their expression levels across our different genotype comparisons. And indeed, it turned out E2 microglia overexpress these phagocytosis genes compared to both the E4 genotype and the E3 genotype. And those were kind of the only comparisons that were multiple testing corrected significant. We went back to in vitro models to test this functionally as well. And so we did this with 2 different phagocytosis assays. So E. coli particles shown on the left and myelin on the right. And when analyzed quantitatively overall, we showed that indeed, in particular in the E2 versus E4 comparison, the E2 microglia are better at phagocytosing both the E. coli particles and the myelin. So overall, each microglia is able to take up more E. coli particles in E2 compared to actually E3 and E4. whereas for the myelin, it's that a higher percentage of microglia are phagocytosing the myelin in the E2 compared to the E3 and the E4. And there is also one really strange thing going on with the knockout microglia that seem to be stuffing themselves, gorging themselves on myelin, so internalizing a lot of myelin that they are not breaking down then. And so we didn't follow this up in this particular study, but it's certainly something to highlight and maybe for future research. Beyond chemotaxis, proliferation, and phagocytosis, another pattern that we observed in one of our modules was differential immune regulation across the ApoE genotypes. And in particular, it seemed that pro-inflammatory cytokines and chemokines were really upregulated in the E4 and downregulated in the E2 genotype. And we're showing some examples here. So as you can see, generally the first three seem to follow this trend, whereas the CXCL16 kind of shows the opposite trend with the highest expression in E2, lowest expression in E4. Now this is an interesting cytokine because it's in the periphery, it's known to be pro-inflammatory, but has been reported to have anti-inflammatory effects, particularly in microglia in the context of brain tumors. And so we do think these results are quite consistent, and what they're telling us is that there's this graded pro-inflammatory immune response that's lowest in E2, intermediate in E3, and highest in E4. And some of that is actually consistent with work reported by Alison Goad's lab a few years ago in the mouse. So they only studied E3 and E4 microglia, just mouse in a mouse model, but showed that the E4 microglia expressed higher levels of pro-inflammatory cytokines. And so these are all really interesting gene regulatory networks and patterns that we detected in our data that suggest functional differences between these microglia. But what we're really also interested in is what might be driving these broad transcriptional responses. So what are the upstream regulators of the responses? And to figure that out, we often use transcription factor motif analysis. So we check whether the regions that are epigenetically different, so where the chromatin is altered, whether they are enriched in the motifs of any known transcription factors. And so indeed, when we looked at those regions that have enhanced open chromatin, so more open and accessible chromatin in the E2 microglia, one of our top hits for transcription factors is the vitamin D receptor, VDR. So this is really, really interesting because VDR is a nuclear transcription factor that, as the name suggests, is activated by vitamin D. And it's interesting because epidemiologically, we know that vitamin D deficiency is a major risk factor for both the developing Alzheimer's disease and also poor disease progression or faster progression once you already have Alzheimer's disease. And so this suggests that in ApoE2, vitamin D receptor is binding more and more effectively. And so that was based on the chromatin data. And then we wanted to see, is it true that also the target downstream genes of vitamin D receptor are also upregulated in E2, as would be suggested by the enhanced binding of the receptor? And indeed, we took a literature-based known target list of vitamin D receptor, and we showed these are upregulated in the E2 microglia compared to both the E3 and even more so compared to the E4 microglia. And one of the well-known target genes is actually IL-10 and the IL-10 receptor, and here shown the expression of the alpha subunit of the receptor, and you can see this increased expression in the E2 genotype. And so overall, what we think is happening is that some of the protective effect of ApoE2 microglia may be conferred through enhanced vitamin D receptor DNA binding, which then downstreams activates these anti-inflammatory cascades via IL-10, IL-10 receptor. And from the literature, we know that downstream of that should be SOX3, which in turn downregulates pro-inflammatory cytokines. And so we're really excited about this result as a potential follow-up. This paper was published just a few months ago now in Nature Communications. So if you want to read more details, I encourage you to look that up. But I also wanted to highlight just this Friday, actually, there's a new paper from Lexi Knott's lab who studied microglia and perivascular cells and showed that, again, AD genetic risk, we know it's in microglia, but this paper also did some drug repurposing discovery analysis and showed that based on the genetics, the sporadic genetics, GWAS genetics of AD in microglia, one of the best target drugs is actually our vitamin D agonist, so vitamin calcitriol as basically the active form of vitamin D to activate downstream vitamin D receptor. So we were really, really excited to see this and sort of these consistent findings suggesting a potential role for a vitamin D receptor as a protective transcription factor in Alzheimer's disease. So moving on, I want to move from just the genetics into what actually happens in the brains of AD patients sporadically, like when you look at their epigenomes. And a lot of that work was started in the DNA methylation field originally, and then sort of histone acetylations came in maybe in 2018, first with our work and then others. But a lot of this work has traditionally focused on bulk brain signatures, and this is slightly problematic because number one, you may have cell type proportion shifts in your sample. So particularly in a disease like Alzheimer's, where patients experience neuronal loss, the proportion of neurons would be lower in a brain sample. And as I've explained to you in the beginning, epigenetic signatures are extremely highly cell type specific. And you don't want to be just picking up that people are losing neurons based on having decreased signals in neuron-specific enhancers. And the second is that, of course, when you have a bulk population of cells, some of the signatures might be swamped out, particularly if you're dealing with a lower prevalent cell type like the microglia. So there's been some advances in single cell and single nucleus approaches that let us look at individual cells, but especially at the epigenetic levels, is basically, in terms of commercial kits, it's only open chromatin ATAC-seq, and this data is still very, very sparse. And so we've been thinking and working on approaches through which we can better get to cell type specific signatures. First, computationally, so we spent some work in the past few years developing algorithms for cell type deconvolution, so basically where you take a bulk brain sample of histone acetylation, and you also have reference profiles from purified cell types. And you can then deconvolute that bulk signature into its constituent cell type specific tracks and signatures. And you can use that to understand what's happening cell type specifically in different diseases. And this work was led by two talented bioinformatics. So Kitty, as I said, was a PhD student in my lab, and Yuki actually originally a master's student and then a bioinformatics research technician. And together they developed two complementary approaches to do this. I'm not going to talk through the maths of it, but basically we have two ways of estimating proportions of cell types. This was using one of these algorithms based on matrix factorization on my original ChIP-seq study from 2018, which was conducted on bulk tissue. And just even looking at this visually, what you can see is that the blue bars are shorter on the left than they are on the right. And that's very reassuring because the left is the AD group and the right is the control group. And so we even just visually see this loss of neurons in AD compared to control. We did a whole bunch of work validating that these cell type proportion estimators actually work. So this was based on pseudo bulk samples, which we had sampled at known proportion of the cell types and then re-estimated the cell types with the estimator, and just to show it works extremely accurately. And I think it works so accurately because enhancers are so highly cell type specific. So we can basically leverage the fact that they're so cell type specific to make these deconvolution approaches work really well. So the ultimate goal, though, was to understand what's going on cell type specifically in disease. And so when we did this on our Alzheimer's disease ChIP-seq study, we did actually, in fact, see alterations in microglia quite prevalently, but then also quite an overwhelming amount of signal coming from the oligodendrocytes that are both up and down regulated in disease. And This did at first surprise us, but there have been multiple other papers since that have implicated oligodendrocytes epigenetically, transcriptionally in Alzheimer's disease, including from Li Wei Tsai and Dervis Sally in recent years. So that's something that we're also interested in following on. But basically what we see is the genetics really very much points to the microglia. And if you look in sporadic brain, you also see microglia signatures, but you also see these really strong oligodendrocyte signatures. So that's the computational approach to cell type specific regulation, but we've also been working on experimental approaches to do this. And so we paired up with Jonathan Mill and other investigators at the University of Exeter, leveraging the Brains for Dementia Research Initiative in the UK. So this is a network of different brain banks to make brain tissue collection consistent and standardized and leverage it as a resource for research. And so we have a joint project where we're using postmortem brain tissue from the dorsolateral prefrontal cortex of AD cases and controls. We're using FANS, so flow cytometry, to purify nuclei of different cell types, so oligodendrocytes with SOX10, microglia IRF8, neurons NUN, and we're actually also profiling a triple negative population, so everything else that's negative for NUN, SOX10, and IRF8. And then downstream, we're performing multiple gene regulatory assays, so including DNA modifications, chromatin accessibility, and H3K27 acetyl. And as you can imagine, my lab is in charge of the H3K27 acetyl, and this time we're doing it with a slightly more novel technique called cut and tag that was developed a few years ago and that we've been optimizing in the lab. And so this really quite big research project was led experimentally by my former postdoc, Joy Ismail, and all the data analysis was done by my PhD student, Paulina Urbanovic-Schulte. So this is where we're at after many years of purifying cells, or nuclei rather, running many cut and tag experiments, up to 600 different cut and tag experiments over the course of a year, and then doing really extensive pre-processing, quality control, and filtering. So this is basically our final data set post-QC with everything removed that shouldn't be there. And as you can see, our samples come from 5 different brain banks across the UK. They're older aged because they're AD and matched samples. They're relatively they're sex balanced basically, and we have about similar numbers of AD cases in control. And so per cell type, the numbers vary slightly because there are just a lot more oligodendrocytes and neurons available. And so in those cell types, we have up to 140 samples in each, whereas in the microglia and the triple negative population, we have around 70-75 samples after all the QC. So when we look at the data overall, we see that the biggest axes of variation, as would be expected, really come from the cell type. So this is the PCA plot of just the overall axes of variation. And you can see that it segregates really nicely. So along this axis, you have microglia against everything else. And along the bottom axis, the neurons versus the oligos with a triple negative sort of almost distinct, maybe bleeding slightly a little bit into the oligodendrocytes. And we think that this triple negative population, because it's depleted of all these 3 cell types, so it should be primarily enriched for astrocytes, endothelial cells, and anything else that's in the perivasculature. And so I'm cutting straight to the chase and giving you the information on what is then actually going on in Alzheimer's disease brain. So we did the differential analysis, controlling for various quality control and batch, sex, age factors. And this is the overall result. And actually, you can see here again that the largest number of differences are identified in the oligodendrocytes, again, as predicted by our computational deconvolution approach. Of note, microglia and triple negative do have only about half of the sample size of the other populations, and so we would expect to have less power in those cell types, so the number of differentially acetylated genes aren't directly comparable, but neurons and oligodendrocytes have similar numbers of samples, so here we do detect more in the oligos, and then in the microglia versus triple negative, clearly detect more in the microglia. So since this is the microglia webinar, I'll focus just on the microglia results and give you some overall highlights. So this is the volcano plot of all the significant genes, and even by just annotating some of the top genes that we find differentially acetylated, you see that certain things stand out. So number one, we see a number of different peaks that are annotated to genes of the type 1 interferon response. So interferon alpha in particular, different family genes from the interferon alpha family. And those are very consistently downregulated in the AD samples. This is just one example. And conversely, one gene that pops up a lot in our upregulated peaks is PTPRG. So this is protein tyrosine phosphatase receptor type G. It's really interesting in the context of Alzheimer's because it has been genetically implicated in Alzheimer's disease through family-based studies. It's also a gene that is consistently seen overexpressed in sporadic AD cases in the microglia in single nucleus RNA-seq data sets. And so it's interesting that we see basically the consistent signal at the epigenetic level as well. And this is a gene for which actually we know some of the mechanism through which it confers AD risk, and that's essentially by activating this M6A methyltransferase called VERMA, which then interacts with mitophagy-mediated neuronal death. So again, these were just a few highlight genes, but if we look overall at the pathways that are altered in AD microglia, and this is the downregulated pathways, there are a few highlight things. So like I said, there is not just in terms of the 3 top genes or so, but we do actually, as one of our top processes, see type 1 interferon receptor binding downregulated in AD. And these are all the genes that are annotated to peaks that are contributing basically to that pathway enrichment. So you can see it's numerous different in type 1 interferon family genes. And the second thing that we thought was really interesting is, and this pops up mainly in the upregulated pathways and maybe a little bit in one downregulated pathways, is basically pathways relating to lipid and phospholipid binding. So on the upregulated side, we see more of the binding and the transfer, whereas on the downregulated side, it's maybe a slightly different aspect of that process, which is the phosphatidylinositol kinase activity. So I'm not sure if these are compensatory feedback processes that drive opposing results, or whether it's really different aspects of that, those lipid metabolism and lipid processing functions are altered in opposing ways in disease. But certainly, especially in light of the GWAS enrichment that I showed you in the very beginning of microglial processes that are implicated by GWAS results, really pointing towards lipid metabolism, it's really interesting to see this confirmed in AD samples at the epigenetic level in just sporadic cases. And then also interesting potentially in what I've told you about the ApoE microglia and the chimeric mouse, we actually see an upregulation of cell motility and cell migration at the epigenetic level in AD cases, and that could be a compensatory response to the pathology. And then, of course, we're always interested in what is driving these responses. So we also did some pathway enrichment analysis, and I'm just going to touch on 2 pathways that really stand out for me as particularly interesting. And number one is this BCL11A that comes up as most significantly upregulated in our AD microglia. This is a transcription factor that's known to regulate microglial immune responses. It's elevated in neuroinflammation and also elevated in AD mouse models. And it has a number of downstream target genes that are associated with AD in our data as well. And importantly, a lot of these PTPRG genes that we see upregulated in terms of acetylation predicted to be driven through BCL11A binding. And the second one that I'm really interested in is this SREBF2. And now this is a gene that's involved in cholesterol synthesis, which downstream has consequences on inflammation and antigen presentation as well. And it's been linked in the past to ApoE4 microglia in AD. So it's interesting, again, also in the context of lipid, to see that come up in our data. And then finally, even though we have purified cell types in this data, of course, when you go into single cell data sets, you have an even more refined substate, subcell type kind of annotation of microglia. And so we actually took some of because we can annotate our peaks to genes, we can ask, are the genes that are upregulated or downregulated in our data, do they overlap known marker genes for different microglial state? And this is from a single cell study from Li Wei Tsai's lab, Sunatel 2023, where we're annotating into the different microglial states that were reported in that data set. And so we see what is actually downregulated in our AD data set is the homeostatic microglia, whereas significantly upregulated we see most strongly actually lipid processing microglia, so very much in line with what we see at the pathway level. And then also this inflammatory type 3. Now, as you can see, they have 3 inflammatory types of microglia in this data set, but essentially inflammatory type 3 is the most extreme. So they see it as a pseudotemporal trajectory that goes from 1 to 2 to 3. So 3 is really end stage super pro-inflammatory microglia. And so what's interesting is that this suggests that we probably have an increase of lipid processing and inflammatory type 3 microglia in the AD samples and a decrease in homeostatic. But on top of that, these signatures are also enriched in the early ADDGs from the same subtypes. So it's not just an increase in the proportion of these cells, but it's also an increase in the expression of the characteristic genes that define these cell types and of the genes that are altered within that subcell type in early stage AD. And yeah, so that's nice in 2 ways, because one, we have converging evidence from the gene expression and the epigenetics, and 2, our late stage samples here are showing basically overlapping genes with the early stage differential expression gene. So we can potentially still pick up early events at the epigenetic level in our data. I am conscious of time, it's already 1:45. I did have a second section on Parkinson's, but I think probably it's better if I just skip to the end and then we leave plenty of time for questions just on this AD part of the talk. So I'm just going to run through to the very end of my slides. And I'd like to thank you all for listening, for the invitation again. Certainly thank all the lab members that did all the hard work on these projects and led these projects, our collaborators who were involved in this. So the things that I mentioned today, particularly Renzo, Bart, John, and Alexi, and all the funders that have been funding this work.
And thank you very much, Sarah. It was an amazing talk with an amazing amount of information. So I think it was a good choice of a decision to stop there. So we're going to leave some time for people to just raise your hand or type your question on the chat.
So we have already a question in the chat. Do you want to ask that? So there is a question from Gloria Colombo. Thank you for this very interesting talk. Since ApoE4 is known to increase the risk of AD in women more than in male, I was wondering, do you see a sexual dimorphism in terms of macroglial epigenetic responses in your model or in your data set?
Yeah, that's a very, very good question. So not so much in the xenotransplantation. So the iPSC microglia that were xenotransplanted were actually a male male background, whereas the animals, I think, yeah, no, actually the animals were also male. So we do pick this effect up in males. We would potentially expect this effect to be even stronger in females. And actually in the human data that we now have, even though as you go into sex stratified and ApoE stratified analyses, you kind of lose sample size, of course, but it's one of the biggest cell type specific epigenetic data sets. We do have genetic data for all the samples, so we do have ApoE genotypes. And in fact, we have been looking a little bit at that. So that's something that we're definitely really interested in. And I don't have any highlights to report yet on that. That's still on the to-do list to dissect that properly.
Thank you very much. Rosa, yes, you have a question?
Yeah, I may ask directly. So thank you so much, Sarah, for a beautiful talk. So I was actually very intrigued by this interesting difference between the genetic risk variance in microglia, which is pretty strong, and the epigenetic that you showed, oligodendrocytes really rich. Do you think so? What is the trigger? Can it be driven by microglia? Do you think it's really upstream microglia affecting? And if this so, how it's happening?
So yes, you touch on a very, very good point. I think there are now consistent, suggestive at this point, pieces of evidence that do in fact suggest that it might be the microglia that are activating the oligodendrocyte. So part of that was shown in the Graham et al. Paper from Dervisali and John Hardy based on the transcriptional network. So they predict that it's actually the microglia that activate the oligodendrocytes. And we've been doing some work in collaboration with Dervis and also Alexi Knott, looking at oligodendrocytes in AD and in aging as well. And what we really see is that the pathways and the genes that are most altered epigenetically tend to be those that communicate directly with microglia and that receive input from microglia. And so, yes, that's my hunch as well, that microglia are going to be a key player. And we want to do some more work dissecting some of the existing single cell data that's out there and potentially adapting some of the cell-to-cell communication computational methodology to apply that to epigenetic data sets as well, to then dissect that a bit more. And maybe eventually we'll have to go into spatial approaches as well.
Super interesting. Yeah, thank you.
Is there anybody in the...
There's another person in the chat. And then I will also have a question.
Okay, Gilbert Paolo asked a question. Fantastic talk, Sarah, and hi. The ApoE knockout versus ApoE4 comparison is interesting. It looks as though the ApoE knockout resembles the ApoE4 genotype in some assays, whereas in the other assays, it resembles the other ApoE genotype.
Yes.
How do you interpret this?
That's 100% correct. I don't know what our expectation was when we went into this, but certainly it wasn't something like ApoE4 is always like the result isn't ApoE4 is always like the knockout. Sometimes it is, sometimes ApoE2 is like the knockout, sometimes both of them are different from E3, so it's not very consistent. We did do overlaps into what is altered compared to ApoE3 in the knockout, in the E4, in the E2. The strongest overlap in that sort of, if you compare the effects, directions and sizes, is between the knockout and the E4. So it seems like the E4 has more of a loss of function, potentially protective function. But some of the protective effect of the E2 is also loss of function. So it's a little bit in two different directions, and it might be affecting different processes. So for example, with cell motility, probably negatively affected, so loss of cell motility and phagocytosis in the E4, that's mirroring the knockout, but other processes where E2 has loss of function, and the loss of function may be protective.
Thank you.
Thank you.
Hey.
Amanda, if you want to ask your question, there is someone else then raising
Yeah, go ahead then.
Yes, TS Kao. If you want to ask your question, I know you raise your hand.
There's another question on the chat.
Yeah, in the meantime, I think I will take the question in the chat. So thanks a lot for the insightful presentation. Regarding the different ApoE genotypes of microglia xenografted into the mouse model, if I understood correctly, you analyzed these cells after plaque formation, around 12 months.
Yeah.
Since microglia might also play a role in feeding the plaques, would it make sense to analyze sample before plaque formation as well?
Definitely it would. So the honest answer is it was basically a money and time limitation. We had to go for one experiment, and we decided to go with this quite high pathology, extreme late time point, and we do see lots of differences, and so that's interesting. But certainly obvious follow-up work would include understanding how microglia are involved in the early or even pre-aggregation stages, and as you said, involved in the seeding process. Something we'd like to do. The xenotransplantations are a lot of hard work, so we're trying to take some of this maybe into a simpler in vitro iPSC-derived microglia model, and trying to see if we can elicit some of those differences and responses in vitro as well, and then also start to maybe pick up the functional differences that we predict based on the gene regulatory data, and potentially start to manipulate the vitamin D receptor and its binding to see if we can rescue some of the protective, so basically push the E4 microglia into a more protective state by enhancing vitamin D receptor binding.
Amanda, if you want to ask the question.
No, I can go. I can be the last one. It's okay.
Okay. So there is Jen Woodward that asks, amazing talk regarding CHCHD2. Have you looked at this in any more detail? Did you get many other mitochondria-related genes?
Good question. Not off the top of my head, any of the main sort of regulatory networks and pathways, not so much. I think it just stood out to us as a Parkinson's associated gene. But then again, if you look at the functions of CHCHD2, it's so pleiotropic. So it is a mitochondrial gene, but it can also, for example, act as a nuclear transcription factor in hypoxic conditions. It's involved in cell motility, both in sort of early development, but also in some chemotactic processes. So we booked it down more as in line with these chemotaxis cell motility differences that we see across the E234 microglia. So it was consistent basically with those signatures, meaning that loss of CHCHD2 in E4 could be contributing to some of that poorer cell motility that we see. But like, yeah, it has so many different functions that it would surprise me if it wasn't in other ways also, and potentially through mitochondrial metabolism and regulation, also involved in causing disease vulnerability.
Thank you. There's another question in the chat, I think multiple questions. So one from Lia Casillas Rodriguez. Great talk, thank you. Were you surprised to see a decrease in interferon receptor binding? Any clues as to what upstream regulatory factors are driving this?
Yeah, no, we were a little bit surprised because, I mean, the simple thinking would always be everything activated and pro-inflammatory is happening in AD and everything anti-inflammatory is controls. That doesn't seem to be necessarily the case, whether this is a sort of a compensatory response, or this might be even a sign of, because our samples, I should have mentioned, I think it was on the graph, but they're relatively late stage AD. So we recruited Bragg stages 5 and 6 into the AD category. And so it could be actually that maybe they did have more interferon signaling in the earlier stages, and then they just get exhausted. And so they eventually downregulate epigenetically in those. We haven't dissected that fully yet, and we are looking for upstream regulators. So these transcription factor results that I showed you are basically just fresh, a week old or so, because we, it took us a really, really long time to do the quality control, the p-calling, the differential analysis really well, because basically this was the first time that we fed a data set as big into the kind of statistical approaches that are used here. And so we were pushing the boundaries a little bit of what was possible and what was not, and had to refine that quite a lot so that we can be confident of our results and see that they're robust as well. So that took a long time. But yeah, that'll be one really interesting question to answer, what lies upstream of those downregulated interferon responses.
Thank you. Another question from Fabia Filippo in the chat. Great talk. I was curious to know, how do you interpret the downregulation of the interferon response?
Yeah.
So it's a similar question.
A similar question. So yes, exactly. That surprised us a bit. I would say in the literature, it's not entirely consistent. So you do see reported in both ways. You see some studies showing upregulation, some studies showing downregulation. And I guess the key in understanding this is maybe figuring out the temporality. Maybe it's the first upregulation in early stages and then exhaustion and downregulation. That would be maybe my strongest guess, but yeah, we certainly need to dissect that further.
Another question from Anandita Banerjee. I hope I say it well. Amazing talk regarding the vitamin D receptor slide. There is a binding with this in the absence of vitamin D, if I understood correctly. Then in that case, women tend to have generally lower level of vitamin D. Do you think it can be one of the major reasons for AD in females?
So no, we don't think there is a binding in the absence of vitamin D. So we do think vitamin D is required for the activation of vitamin D receptor generally. And in fact, it's not just the vitamin D, but the receptor actually heterodimerizes with a retinoid X receptor before it binds at vitamin D response elements in the genome. And in fact, one actually confusing finding is that often ApoE4 microglia have been shown to actually contain a lot of vitamin D, so they're kind of almost fuller of vitamin D, but they might not be using it as effectively. So maybe it's the activation and the downstream binding of the vitamin D receptor that is inhibited or not working as well in the ApoE4 microglia. And we are certainly interested in figuring that out in a bit more detail. And understanding what is required and where is this going wrong, because I don't think it'll just be vitamin D supplementation. And I also think this is why we don't know, but supplementation trials in the past have led to mixed results, let's say. Some are successful, some are not. And so that suggests it might not be as simple as giving vitamin D and saving things, but it might require a number of other things. Could be, for example, the function of retinoid X receptor.
Thank you.
Yeah,
I think you can.
Yeah, okay. Just one quick question, because it's already 6 p.m. But going back to your previous question, I was wondering how, I'm not sure if you really tested it, so it's more like a speculation. How permanent do you think the signatures are? Like, and my question is actually, when do you think they arise? And whether you can use them for predicting the evolution of the disease. How stable they are, do you think?
We don't know, and the problem is that for each individual study, we basically only have one cross-sectional time point, right? And often we have, for now, focused on late-stage brains just because we wanted to increase our power to detect anything, because with the argument that if you take this really late-stage disease brain, you're more likely to pick something up, just in case there's it turns out there's a lot, almost too much signature. And so, yeah, some of our follow-up work and some of the grants we're writing now certainly goes towards trying to dissect earlier temporal stages of disease as well. What do I think, how stable they are? Well, I think the fact that we see some overlapping signatures with the early change differentially expressed genes in the Sunadal paper in our epigenetic late stage signatures, that's that gives me hope that even in the late stage you can detect things that are maybe relevant to the earlier processes that drive upstream, potentially causally, Alzheimer's disease in these samples. But until we study them, we can't be sure yet.
Okay. So we have, I don't know, Rosa, do we have time for one more question? We have one more in the chat.
Yeah, maybe the last. We can get the last.
Very last one. Okay.
So we have, hello, thank you so much for the talk. Given that many AD genetic risk variants act through macroglia-specific regulatory elements, and that several lipid-related pathways are upregulated in AD microglia, do you think future therapeutic strategy could focus on modulating these upregulated genes or pathway to restore microglial homeostasis?
Yes, data would certainly suggest that this would be a beneficial thing. I think there's quite a lot of evidence as well that these alterations in lipid and cholesterol metabolism might come upstream of inflammatory, potentially toxic inflammatory responses as well. So that's certainly the case. Now, I'm not a lipid expert, so it's just my data and our analyses that have sort of repeatedly led us to this lipid result. And so I still have a lot more to learn, but we're starting conversations and collaborations with labs that have been a bit more focused on lipids. And so we're excited to see where that will go as well.
Great. Well, it was an amazing talk. We had a great amount of questions and very interactive sessions. So thank you, everybody. Thank you, Sarah. And we'll see you next month. Let me remind you that it will also be on Monday.
Yep.
Okay. Thank you very much, everybody.
Thank you.
Bye.
Bye. Bye. Bye.